we have already studied about animal husbandry methods. Now let us take up the agriculture sector. We know that India is an agricultural country. More than 60% of its people are engaged in agricultural practices. And with the increase in food demand due to increase in population, initially traditional farming methods were integrated with good management practices. But there was very little increase in yield. So, in 1960s, a new concept of green revolution was introduced in our country by adopting better plant breeding methods, which resulted in drastic increase in the yield. What are these methods of plant breeding? In plant breeding methods, the high yielding plants are crossbred with the disease resistant varieties to get a better breed. If you see this flow chart, from very long time, that is around 9,000 years ago, this practice has been done, which is called as conventional breeding. In conventional breeding methods, the wild varieties are used for cross pollination to get a better yield, or the two asexually, the grafted varieties are also used. But this conventional breeding method, though it is good to increase the production, but it has some limitation. So what is the need? The need is to adopt new methods which are called as classical plant breeding methods, which includes mutation breeding and tissue culture methods. So these crossbreeding methods include artificial selection of good varieties of crop. So if you compare any crop, there are certain good agronomic traits like high yielding quality, tolerance to water scarcity, salinity, or resistance against the bacterial, viral and fungal diseases, or resistance to insects. So plant breeding methods includes researching so that these agronomic traits can be inculcated in our crops. So these are called as plant breeding techniques. These are the steps of plant breeding technique. The first step is the very important step, that is called collection of germplasm. In this, all different variations of that particular crop and its wild relatives are collected for the breeding. This is called natural gene pool. And from this natural gene pool, depending upon our requirement, we select the superior parents. These superior parents can be for any agronomic traits. So these agronomic traits are selected based on the requirement, and then these pure lines of these superior parents are developed. After that, there is a cross hybridization of these superior parents. You must be remembering artificial hybridization. So artificial hybridization technique is used to get a superior hybrid. That hybrid which has both the characteristics of the parents. That is called as hybrid vigor or heterosis. So whatever hybrids we get, selection of that superior hybrid is a very important step, because it's a tedious step. All the hybrids which we get might not be of our desirable quality. So selecting the superior hybrid is very important. After that, these selected superior hybrids are tested for their relevance, for their potency, by growing them and producing the pure line of that hybrid, so that this can be further evaluated for its traits. And after evaluation of these traits, then the scientists release them as a new hybrid. and these hybrids are cultivated for at least 3 seasons to see the response, and when the response is desirable, then it is compared with the best available cultivar of that particular area, and when it is successful, then only it is released as a new hybrid. So plant breeding methods have been very effectively used in most of the crops like rice, sugarcane, wheat, as well